Now this lecture is basically culture type based on its sustainability, means like we are talking about the different cultures and how these cells are sustained on the culture. Sustenance of the culture is basically depending upon the nutrients requirement, nutrients provision, and these nutrients concentration also. And if you go back again and compare to the different types of media, differential media, enriched media, selective media, synthetic media, defined media, complex media, all these media, whether they are solid form, they are liquid form, they have certain limit of the nutrients to provide and to sustain this bacteria. Now, in the last lecture, we talked about the continuous culture. In the continuous culture, we discuss about the growth of the cell will be maintained and sustained by providing the additional nutrients or additional supplements over a period of time. All right. So this is how to maintain the sustainability. We'll discuss in this lecture. Now, if you talk about the batch culture, right? So there's a difference between continuous culture and batch culture. Batch culture is something where you inoculate and it will reach to certain point of time and then the growth is exhausted, right? So batch culture is closed system like in tube or in flask or in fermenter, where the microorganisms grow in a fixed volume, 10 ml, 100 ml, 1 liter, 10 liter, 1 gallon, 10 gallon, right, in a specified volume of a medium without addition of a fresh culture nutrients. Now, when you don't add the fresh culture nutrients, culture nutrients means like nutrients which are required for the culture maintenance, then it will reach to a certain level, certain stationary level, and then the product get exhausted, right? Now, this is the most common laboratory cultivation method. So the fresh nutrients are the removal of the product takes place. This is how one batch. Similarly, you can have another inoculum. Again, you go for 10, 20, 30 hours in a specified volume to make another batch. Then third batch, fourth batch, and accordingly you process these batches for the purpose that you are making. Maybe for protein isolation, for DNA isolation, or for whatever purpose. So inoculation first, growth phase, and the resources depletion comes, and then you can get the product. Product means like whole culture will be taken out as a product. Now, batch culture growth phase. Again, you have a lag phase, exponential phase, stationary phase, and death phase. So, cell adopt the conditions and acclimatize to the situation in which they have been grown for a few hours, a few minutes, depending upon the type of cells and type of the nutrients provided to them. So, cell adapt a new environment, synthesize enzymes, no net increase in the cell. So, this is called the lag phase, as I have already defined in the growth curve earlier. Then exponential phase comes. It increases to the logarithmic increase of the cell. Then stationary phase comes. In the stationary phase, the cells exhaust to grow because of the depletion of the nutrients. Then, of course, when the nutrients are completely exhausted, then the death takes place. Now, what is fed batch culture system? Fed batch culture system is a sort of transit form of or intermediate form of the continuous and batch culture. In continuous culture, on the basis of the turbidity or chemical composition by measuring the turbidometry or turbidostat or what you call turbidostat and the chemostat, right? You can measure the level of the nutrients depletion and you can add accordingly the required nutrients so that the growth continues and it will remain on the exponential phase, right? Or at least at the stationary phase. It should not come to the decline phase. But in the batch culture, it is not like that. It comes to the stationary phase and then come to the decline phase. Whereas in the batch fetch culture, which is intermediate state of intermediate form of the culture, is in which it involves the periodic or continuous addition of a fresh medium. So, for example, after 10 hours or after 8 hours, you find the growth get stationary. our growth get to the kind of a intermediate decline phase, then you add another culture media, another fresh media, so that the culture can enhance and again grow the cells, more cells in the exponential phase or in the live state. So nutrients addition, fresh media addition, periodically you can standardize after 2 hours, 3 hours, 4 hours, whatever. Once the cell reaches to the exponential phase, then you have to know after what is the time required to deplete the nutrients and exhaustion of the cells to further grow. Then extend the growth. So maintenance of this exponential phase longer than the batch culture, because in batch culture you can have, for example, if in a batch culture you have like this at this time, but in case of fed batch or continuous culture, it may go to up to continuous on and on and on, because at every point of time you are adding 2 hour, 4 hour, 6 hour, 8 hour, you are adding the fresh media, you are adding the fresh nutrients, supplements, so that the continuous growth, exponential growth may takes place. So this is called the fed culture system. Now the fed batch application. What is the fed batch application? If you go back to the continuous culture, as we discussed on in my last lecture, there we talked about the industrial use of the continuous culture. Similarly, the fed batch culture is also used for the industrial purpose. Industrial production, for example, protein factors, say for example, insulin production. So if you have an E. coli with the gene of insulin production gene, recombinant E. coli cells, clone, and you grow these clone for the production of the insulin, then insulin will be produced through fed batch system. Because in that case, you can have a large number of insulin, a large amount of insulin can be produced. So the yield of the insulin can be enhanced, and this yield when enhanced, then you can have a better quality insulin produced from this bacterial population. All right. Now, so fed batch culture has advantage over the batch culture. So industrial production is one, then metabolite control is another one. So industrial production widely used in biotechnological industries, antibiotic productions, enzyme production, recombinant protein production, growth factors, protein. So in case of metabolite control, it allows the control of a specific nutrients to direct metabolism. Right? For example, if you have a specific cells to grow and you desire some specific product, so accordingly you have to provide a specific metabolite, specific nutrients to be utilized by the cell and to produce particular metabolite. For example, if you are going to produce a bacteria used as a bioindustry for the plastic productions or biodegradable plastic production. In case of biodegradable plastic production, what you need are specific nutrients provided to the cell so that the metabolism of the bacteria utilizing that nutrients can provide that plastic. Right, and then you can keep growing. So keep providing that metabolites, that nutrients to the bacteria in order to grow or to physiological active bacteria to grow the plastic and set, so that you can, after a period of time, 10 hour, 20 hour, 30 hour, or even overnight, you can get the ample amount of plastic, biodegradable plastic to be isolated as a product. So this is metabolite control. You can also control certain metabolites. You can also inhibit these some metabolites by providing certain toxins, certain nutrients which can inhibit. For example, you can provide certain nutrients in order to inhibit certain metabolites. So if you want to get the bacteria growth and get the product having depletion or decreased amount of a particular metabolites, so you can also control that from fed batch culture by providing that specific antitoxin or that providing specific toxins or any molecule that can inhibit the growth of a particular thing. Now, what is continuous culture? Continuous culture maintain the microorganisms in exponential growth, right? It means the growth, logarithmic growth is continuing, growing without any loss of growth, without any reaching to kind of a stationary phase or the decline phase. Now, the two different type of methods are required to control the continuous culture. The chemostat, that is a nutrient limited, right? So you can have the nutrients can be calculated, can be estimated, can be measured. Accordingly, you can keep adding these nutrients in the, so through chemostat you can identify the nutrients. Then turbidostat. Turbidostat is basically to control the turbidity. Turbidity means like the growth of cell. By looking the growth or the density of a cell, right, you can check through turbidometer, and you can say, okay, cells are growing more. So at this concentration, at this turbidity, cells are more and more, and they may reach to the stationary phase, decline phase, and lost the exponential phase. So in order to maintain that exponential phase, again you have to provide the additional this media. Now, chemostat operation, how to operate the chemostat? Fresh media input, right? So there's a specific setup of the chemostat where you can have some flask, something which can added in this flask through outside, an inlet and outlet system. From inlet you can keep adding the specific nutrients media, and from outlet you can keep extracting the growing cells which are in the exponential phase or any required phase that you want for any research or any product development or any growth of a particular microorganisms. So fresh medium input, sterile medium continuously added at the controlled rate. Now, why it is the controlled rate? Because if you don't provide the controlled rate, then the media may be disturbed and it may either fast grow or either it may slow grow. So you have to have a very controlled nutrients providing in order to control growth. Controlled growth means one nutrients limit growth rate. Right? For example, if you have say for example some antibiotic or some growth factor, so you should keep the whole media providing the single supplement provide or control the whole growth of the cell, right? So maintain the steady state. Steady state means like cell density and growth of the culture remain same. Now, if the steady state is lost, then you may have loss of the continuous culture. And the best example to explain is the synchronous growth. I will talk what the synchronous growth is. Then you can have a removal of the culture. So culture fluid continuously removed at the same rate as input. Now, growth kinetics fundamentals. Growth kinetic fundamental means what are the different growth factors. For example, mu we call the specific growth rate. So rate increased in a cell number per unit time per cell. Then we call TD, doubling the time. Doubling time means like a 2 to 4 to 8, 16 and so on. Time required for cell population to double. So this is also called generation time in growth curve rate. Then we have a yield coefficient. Yield coefficient is amount of biomass, right? Biomass product means like the metabolites and the byproduct produced in the bacteria. It could be whole bacterial mass, protein mass, exclusively some nitrogen compounds. It could be some bioproduct, right? So it could be anything, dry weight of bacteria through this yield coefficient. So these growth kinetics are used to look into the growth of bacteria by calculating the TD, doubling time, specific growth rate, and then yield. And growth rate and generation time is inversely proportional, as we all know. It is inversely proportional. The mathematical model of growth. What is the mathematical model of the growth? So this is a simple equation where you can calculate the through these coefficient, right? Mu, TD, and Y. And just by calculate this total growth and initial growth, epsilon to the power mu and t, and this formula will calculate you mu maximum and then k kinetic. Now this kinetics will tell you the whole growth rate, how fast the growth taking place. Now this can specifically be identified for specific metabolites. Now the real application for this kinetics or growth kinetics, or the model for the growth kinetics is, for example, insulin production or polyhydroxybutyrate, or for plastic production. What you have to do, you have to calculate at different time period, a different timeline, what is the production, total output of these metabolites. So that can be calculated through this mathematical model. What are the factors which are affecting the growth kinetics? So temperature is one, then pH, nutrients, oxygen. Temperature is obvious. I mean, it depends on what bacteria you are using. Generally, mesophiles are used for that. So in case of mesophile, 37 degrees centigrade temperature is required. Now, in case, suppose if you have a temperature sensitive mutant or temperature resistant mutant. So in that case, you may have fluctuation of a temperature may also hamper the growth of these cells, and it affects the kinetics. It affects the growth kinetics of the cells, and ultimately the production of the yield factor of the that particular product that you are producing through these bacteria. pH is again which influences the enzyme function, metabolites production, various physiological activities can be changed or are affected by the pH conditions. Oxygen, right? Some of the aerobics, anaerobics, facultative anaerobic, stringent anaerobes, all these different situations of a bacteria or different type of bacteria will be affected through presence of oxygen. Then nutrients, we have a carbon energy production, nitrogen for energy production, phosphorus. So all these nutrients which are provided for the energy production are also important for the maintaining the kinetic or for stable kinetics. What is the application of a growth kinetics? Industrial biotechnology. Now, optimizing the fermentation process, scaling up production system, predicting product yield and quality control in manufacturing. So this is the industrial application of the growth kinetics. Now, how do you maintain the growth kinetics? How do you maintain the time required for the production of this individual product or metabolites, right? It depends on many factors, as we have just talked about: temperature, pH and other stability factors, nutrients, right? In order to maintain the particular required yield, right, we have to have optimize these conditions, factors required for the growth conditions, so that we can have a better product. So optimizing the fermentation process means the growth process or the process where the growth takes place, because ultimately growth of these microorganisms are required for the final product development. All right. Now, if you won't optimize, then it is difficult to get the required or desired product in the desired and required quantity. So this growth kinetics is basically help to understand, right, what is the generation time of the cells, what is the growth rate, right, accordingly what is the output, the product development. So for example, the product development is directly proportional to the growth rate. So this can be optimized, right? Optimization, you can get the better quality of the, so this is one of the applications of the growth kinetics. You can scale up production. Suppose if you are getting the low scale production, then you can scale up by maintaining the growth kinetics. So you can estimate the growth kinetics, you can increase. So faster is the growth rate, faster will be the product development, more is the product development. So this is one advantage. Predicting the product yield, you can get the yield of the product. It's very simple. Now, quality control is again in manufacturing is very important, right? Quality control means also depends on the which product you are developing and what is the precursor or what is the requirement of the nutrients to get the better product, right? So this can also be calculated through, if there's a growth rate is low, growth kinetics is low, then you can replace that nutrient or that for the production for the fast or slow growth of the microorganism. So once you know the growth kinetics, you can manipulate the whole growth of the microorganisms and to get the better quality product. In research application, of course, studying the microbial physiology, how the microbes are grown, what is the environmental conditions required for the growth of microorganisms, so you can monitor accordingly. Drug development and testing. So for example, if you are working on some bacteria for growth, for some growth factors, some drug development or some other things, then you can also use the kinetics to look into what is the rate of the production of these drug molecule, what is the rate of production of these growth factors in the bacteria. So in a nutshell, the growth kinetics is very, very important. Growth rate, to understand the growth rate is very, very important to understand in for a different bacteria. For example, if you talk about the E. coli, if you talk about the Klebsiella pneumonia. Now, growth rate of E. coli and Klebsiella pneumonia and Pseudomonas and A. aureus, S. aureus or A. baumannii, it varies because the generation time is slightly varies. Although it's not much difference between these 2, 3, 4 bacteria because they belong to closely related groups. But if you take, for example, Mycobacterium tuberculosis, Mycobacterium leprae for leprosy, then the growth rate is much different from the Enterobacteriaceae family members or Enterobacterial family members like E. coli, Klebsiella, Pseudomonas. Now, for the research purpose, you have to optimize the growth kinetics. After getting the growth kinetics, optimize the conditions for the growth. Suppose if you find the low growth, you can increase the condition through better supportive media, enriched media. Then you can have a different supplements and nutrients, right? Or you can provide the continuous cultures. Suppose if it is not continuous cultures, you can have a fed batch cultures. So all these things can be made available only and only after you understand the growth kinetics. Now, modern culture techniques. So in case of modern culture techniques, automated system, you have a microfluids, online monitoring. So earlier it was not available. So computer aided or computer controlled bioreactors are available. So you can just monitor the deflection of these needles or in the digital form. What is the level of nutrients? Even you can specify the certain nutrients, the level of nutrients available or not. So the sensors are available. Sometimes nanosensors are present in these microfluidics devices where you can check during the culture growth. You can check the rate of the kinetics of the growth. And so accordingly, you can keep changing the environmental conditions or the physiological conditions for the growth of that microorganisms to get the better product. So online monitoring is also, so real-time monitoring is also possible with these things. Now, future perspectives. What are the future perspectives? Now, AI-driven optimization. So machine learning algorithm optimize the culture conditions and predict outcome. So now on the basis of whenever we talk about the AI, whenever we talk about the machine learning, we must have some database information. So you must have to collect the database information related to environmental conditions, culture conditions, and the growth kinetics. For that, growth kinetics is very important to understand, to study. Now, once you get this data, for example, at this temperature, pH conditions, this is the growth rate. In another temperature conditions and nutrients conditions combination, you have this growth. So this kind of information is available. If then through AI model, through AI driven optimizations can be easily possible. Sustainable practices can be possible. You can sustain the growth of these cells. Environmental friendly media can be developed, can be designed, and energy efficient cultivation system can be developed, right? Precision cultivation. You can very precisely cultivate specific microorganisms through these techniques. So this is the modern era. You can have better prospects of identifying and calculating the specific microorganisms required for a specific growth, all right, and for the specific product development. So in a summary, the take home message is that having the knowledge of culture conditions, right, requirement of the culture conditions and media requirement, nutrients requirement for a particular microorganisms and their product development, right, can be easily understood through all these study through culture conditions, and they maintain this culture for sustainability, growth of these cells through kinetics, through growth kinetics. Now, this growth kinetics will tell you the conditions required to optimize. Optimize means the best quality growth, the best production of the required microorganisms product that you are getting. For example, again I am taking example of insulin. Suppose if you want to get insulin from the bacteria E. coli, recombinant proteins are produced. Now, in one condition you are producing the 1 liter per 1 gallon of culture, for example. In another condition you have a 10 liter per 1 gallon, 10 liter of insulin per 1 gallon of culture condition. Now the culture conditions definitely different from the earlier one than this one, latest one, in which you have high yield. So how did you optimize these conditions? Through understanding the growth kinetics. So therefore, try to connect this growth kinetics with the culture conditions, requirement of nutrients and all. Thank you very much, and I see you again in the next lecture.